I think we're going to get started. It's a few minutes after 1, and we want to be conscious of Dr. Miller's time. So today, Dr. Miller is going to be joining us. He's a professor of pathology, microbiology, and immunology in the School of Veterinary Medicine and an adjunct professor of medicine in the School of Medicine. Much of his research is done using laboratory animals for research on AIDS, influenza, and SARS-COVID to study the pathogenesis of protective antiviral immunity to and test vaccines and immunotherapeutic strategies against these viral diseases. So I want to welcome Dr. Miller and really thank you for being here today. And I'm going to let you go ahead and share your screen. Thank you.
Thanks for the introduction. I appreciate that. Today I was asked to give you guys an overview of SARS-CoV-2 vaccines and where we sit. And I see we already have a question in the chat, and I'll probably get to that toward the end of the talk. But my plan is to go through this fairly quickly and give kind of a basic talk in like 40 minutes or so, and leave you guys at least 20 minutes to answer questions or ask questions, and I'll try to answer them. One thing I wanted to add to the introduction, if you guys know, being UC Davis affiliates, that it's important to acknowledge the centers I'm in, not just the departments I'm in. So I'm also in the Center for Immunology and Infectious Disease and the California National Primate Research Center, just to give the directors their nod as well. So the point of today is to try to give you an overview of SARS-CoV-2 vaccines. And I'm trying to, by the end of the lecture, give you guys a very basic understanding of the virology and immunology of SARS-CoV-2. I understand that there's a wide variety of expertise in biology on the call. So I've tried to make it fit to everybody with something at all levels. I'm also going to explain the organization of the SARS-CoV-2 vaccine pipeline and go over the status of the top SARS-CoV-2 vaccine candidates in the United States anyway. I teach the animal virology course, which is the only virology course, undergraduate virology course on campus. So I'm used to teaching undergraduates, and this is how I start the course. And I think it's a good slide to start with. I can't see my top of my slide, but I know it says everything should be as simple as possible, but not simpler. And that was obviously a quote by somebody a lot smarter than me, as these other two quotes will be too. The second quote is really sums up 2020, and I think everybody would agree with this now. The single biggest threat to man's continued dominance on the planet is the virus. And this was a quote by Joshua Lederberg, who's a Nobel laureate bacteriologist, so he wasn't even part of the team in terms of the virology, but he understood the threat. And then the last quote is, I think, just a great quote, and it's from a Nobel Prize winning immunologist. Peter Medawar won the prize, but Jean Medawar was always a co-author and probably had most of the good ideas. And they said, and they were quoted together on this, they said a virus is simply a piece of bad news wrapped up in proteins. So that's a really good way to start this lecture. And I want to define for you what a virus is. In Latin, virus means poison. And how we define it in biology is it's a very small, infectious, obligate intracellular parasite. And that means a lot, those words. Obligate intracellular parasite is the most important thing that you guys probably, not everyone's familiar with. It means that the virus can only exist or replicate inside a cell. So it has to get inside a cell to make more virus. All viruses have a DNA or RNA genome. So they have a nucleic acid genome, and it can be of the two flavors that nucleic acid exists, either DNA or RNA. And then once that genome is in a cell, it replicates itself. So it makes more genome, and it uses the cellular systems to make more genome. And it also makes protein, and it uses the cellular systems to make more protein. So the new virus that comes out the other side of the cell is completely new. It's synthesized de novo in the host cell based on the blueprint that's in the RNA or DNA genome that the virus inserts into the cell. So viruses are really unique in that they completely cannibalize or parasitize or are molecular parasites on the molecular machinery of the cells. And then once the progeny virions exit the cell, they go on to infect the next cell in the host or infect the next host, and they are transmitted from organism to organism. One of the kind of basic biology ideas to get across before we dive too deep is this idea of the central dogma of molecular biology, which is how information flows in the cell. The body basically functions as a protein machine. All the cells are made of proteins, so we have to have a way to encode all the proteins that make up the cell and then make up the body. So the proteins are encoded in DNA, and that DNA is transcribed into RNA, and we call this messenger RNA. And that messenger RNA is then translated into protein on ribosomes. And this all happens in the cell. This is in the nucleus of the cell, and this is in the cytoplasm of the cell. So I said that viruses can have either RNA or DNA genomes, and these are the molecules we mean. These are the cellular molecules I'm showing here, but the viral DNA is exactly the same as the cellular DNA. It can get inserted into the cell, and once it's inserted into the cell, it makes RNA that encodes for viral proteins, and those viral proteins are then translated on the ribosomes. In the same way, if it's an RNA virus, it just inserts its RNA into the cell, and that RNA is directly translated into viral proteins on the host ribosomes. So the viral genome is really a parasite, as I said, of the host molecular machinery, and it just inserts its nucleic acids as a substitute for the cellular nucleic acids at the appropriate step in the protein synthesis machinery. So there's about 32,000 different viruses that infect mammals. And as far as we know right now, there's about 200 virus species that infect humans. There's about 100 different viruses right now living inside human lungs around the world. Not in the same set of human lungs, but in all the human lungs around the world, there's about 100 different viruses right now replicated. So viruses in lungs and human lungs are not a big deal. Right now, you have viruses in your lungs, you have viruses in your GI tract, skin, nose, and mouth, but most of these viruses have little or no impact on health. When we talk about viruses, to give you an relative idea of the size, we said they're small. What does that mean? This is a figure showing the human hair. This is fine beach sand, grain of salt. These are cells. We're getting into the white blood cells that are in your blood, obviously. Red blood cells, which carry your oxygen. This is pollen. Again, red blood cells. This is a respiratory droplet. So this is what we're all trying to avoid spewing by wearing masks, and of course, breathing by if people aren't wearing masks. And down here, we're starting to get into virus size. So here we have coronaviruses, and then even smaller, we have things like bacteriophages and Zika viruses. So viruses are extremely small. They're much smaller than cells, and you can see that they are well positioned to float through the air in coughs and sneezes and through aerosols and droplets. So in terms of size, about 500 million rhinoviruses can fit on the head of a pin. It's usually a visual most people can appreciate. And when you sneeze, the aerosol from that sneeze and the droplets, not just the aerosol, contains enough virus to infect thousands of people. This is what a sneeze looks like. This is small droplets that are in the aerosol, and they can travel in a cloud through the air for many, many, many feet. We're talking 30 feet, 100 feet, depending on the airflow. These are the large droplets, which we're trying to avoid spreading when we wear masks. And these usually fall out of the air in about 6 feet. But that 6 feet is not a hard number, obviously. As everything in biology, it's a bell-shaped curve. And, you know, depending on the experiments you read, maybe 50 to 80% of droplets fall out of the air in about 6 feet. So this is how these viruses are transmitted, or how they gain access to the air. And the way they're transmitted is you walk into either the droplets or that cloud that's floating in the air, and you inhale a lot of the virus. This is what the virus looks like in cartoon form. Again, it's very small. I showed you how small it was. The main thing I want you to appreciate here is in the inside is an RNA genome, but on the outside are these green spikes that stick out of the virus, and conveniently those are called spike proteins. And you may have heard that the vaccines that we're making are directed toward the spike protein. And the reason they're directed toward the spike protein is in this cartoon, it shows the virion in red up in the left-hand corner here, interacting with the cell in this blue membrane, is the cell membrane. And spike protein binds to a protein, transmembrane protein on the host cell called ACE2. And then that spike protein is cleaved by an enzyme called TMPRSS2. And that leads to a conformational change in the spike protein, which allows the virus to fuse the viral membrane to fuse with the host cell membrane, and then the RNA is uncoated into the cytoplasm. So now we have viral RNA in the cytoplasm of the cell. That undergoes replication to make more copies of the viral RNA. And just like the host RNA that I described when we talked about the central dogma of molecular biology, the RNA is also translated on host ribosomes to make viral proteins. And then after we have a collection of new RNA that's been formed and new viral proteins that have been formed, new virions form, and those bud out of the cell. And that's how the infection is propagated. So this is the genome of the virus. It's a really large genome. I know you guys don't have much to compare it to, but it's a 30,000 base pair genome, which is about 3 times as large as, say, influenza virus or HIV or most of the other RNA viruses we deal with. One thing I wanted reason to show this to you is to show you that this is the spike protein. This is the size of the spike protein over here, this S. So it's a relatively small part of the genome. And the other proteins that make up the virion, the structural proteins, are these other red proteins. But what's really unusual about coronavirus is all these blue proteins, excuse me, my dogs are barking, are associated with control of virus replication. So this is a really unusual virus in that it has a lot of proteins that are associated with control of virus replication. In terms of the genetic relatedness of this virus, SARS-CoV-2, compared to other coronaviruses, this is a phylogenetic tree showing the coronavirus family. You can see there's a couple different subfamilies, sarbecoviruses, novacoviruses, etc. And the important point to understand about this is that this is the SARS coronaviruses that emerged in 2019 in China. And you can see that they're closely related to a number of viruses that are in pangolins and bats in China. And previously we had another closely related virus that emerged from China in 2003, and that's SARS-CoV-1. And you can see that it's closely related to another set of bat viruses that are also circulating in the same geographic region of China that these human infections emerged from. So there is a number of bat viruses that are out there that are closely related to the viruses that emerged in humans. And it's reasonable to suspect that there may be another SARS-CoV-3 out there somewhere trying to enter the human population. The other thing I point out in this phylogenetic tree is the starred viruses are all viruses that now infect humans. We have MERS-CoV, which is a Middle Eastern respiratory disease virus, and these human coronaviruses, which I'll talk about later, which are seasonal coronaviruses which circulate in the human population every year right now, and they have been for a while. So to go over the major milestones in terms of our current virus and its outbreak, as far as we know, it was the first recorded case was on December 8th in Wuhan. And on December 31st, there were at least 27 cases of pneumonia of unknown cause reported in Wuhan. By January 9th, China had identified, so 9 days later, China had identified the novel coronavirus that caused this outbreak of unknown pneumonia. And they'd actually posted the sequence of the virus by that time, by January 11th. By January 13th, the case of a traveler, there was a first case outside of China from a traveler that went from Wuhan to Thailand. By January 20th, human-to-human transmission was confirmed in China. By January 23rd, Wuhan was locked down, like nothing we'd ever seen, like nothing you could ever get away with in this country. By January 29th, despite the lockdown, the virus had already spread to 34 provinces, to every province in China. By January 30th, WHO declared this a worldwide epidemic alert. And then in February, WHO increased the risk assessment, and the International Committee for the Taxonomy of Viruses named the virus SARS-CoV-2, which is what we're left with, basically. It's sudden acute respiratory syndrome coronavirus 2. And WHO named the disease COVID-19, which is coronavirus-induced disease 19. In March, WHO declared a pandemic, and that is, if many of you may recall, but at the end of February, we had our first case in UC Davis, which is the first case of a community-acquired transmission in the United States. By March 11th, WHO had declared this a pandemic, and by March 13th or so, pretty much everyone's life had changed. By October 2nd, we had 34 million cases worldwide and 1 million deaths worldwide. Obviously, that's changed a lot in the period from October to now. To give you a sense of how rapidly this disseminated through the world and where it disseminated through the world, this shows the time frame from December 2019 through June 2020. And you can see that the orange is Western Pacific, so that's China. You can see that there was the outbreak in China. The outbreak in China was snuffed out, but in March, basically, the outbreak in Europe exploded, and shortly after that, the outbreak in the Americas exploded, with the Americas winning the race to being the most infected region of the world from about June, about mid-May on. We've held the crown in terms of the most infected region of the world. So that gives you an idea about how fast things have spread and the relative spread in different regions of the world. This is the daily trends in the number of COVID-19 cases in the United States reported to CDC. This is something I downloaded last week. So it's about a week out of date. I really should have downloaded this week, so I'm sorry about that. I got busy. But it gives you a sense of the what we call the waves. We had our first wave in spring. We had the second wave, if you want to call it that, in summer. And now I would say we're deep into the third wave and possibly starting the fourth wave, as you can all see. And as everyone knows, the hospitalizations and severe cases come about 2 weeks after the increased number of cases. So this is why the public health authorities are freaking out, because we are now at a place we've never been before in terms of number of cases, which means in 2 weeks we'll be in a place we've never been before in terms of number of hospitalizations and number of deaths. This is, again, something I downloaded last week, so it's a little out of date. This is in the United States now. We have 15 million cases. We have over, as you guys all well now, over 300,000 deaths. You know, if you don't download every week, you're 150,000 deaths behind. To give you a sense of how common this is, this whole process. I mean, we all think this, oh, we're living through something new. Well, not really. This happens about every, I don't know, 50 years or so, maybe a little more often, depending on the virus. But in 1918, we had an outbreak of Spanish influenza, which caused a pandemic, which killed about 50 million people. 57, we had an Asian influenza pandemic, which killed about 1 million people worldwide. Hong Kong, 68, killed about 1 million people. SARS, I mentioned that before. That was a very limited outbreak, not really an epidemic. That killed about 1,000 people. The novel influenza in 2009 killed about 500,000 people, depending on who does the math. And MERS, the other coronavirus that's only in the Middle East, or people that hang out with camels, it's killed about 1,000 people. In contrast, coronavirus, as we said, has killed over 1 million people worldwide in October. This is a little out of date slide. So just to show you that we have cyclic outbreaks of viruses. They come from either bats or birds, and sometimes they come from bats to camels into people. But these are viruses that circulate in the wild and regularly are introduced into the human population. In terms of how this virus fits in to the general scheme of things, in terms of its relative severity, this is the relative severity of diseases caused by novel retroviruses. These are influenza A, number of cases, number of deaths. It just, don't look at the absolute scale, but just it's sort of the relative level. You can see that influenza infects a lot of people. It kills about, I don't know, half, a little more than that. In terms of COVID-19, it seems to be a more, the people with, and what we're looking at here is cases of severe disease. The people that get severe disease seem to die more frequently. And with SARS and MERS, the people that get severe disease seem to die more frequently. So in general, the people that get severe disease with the coronaviruses tend to die more often than the people that get severe disease with influenza. And that's one reason the clinicians keep saying, this isn't influenza. And they're right, of course. Pesky clinicians. This gives you an idea about the effects of the relative case fatality rate of these different viruses. Again, it's the same set of viruses. This is by age group. So in terms of the ages, we're looking at more than 70 years old, in their 60s or less than 60. And you can see that in the coronavirus, it's really the older age groups that are most severely infected. The people, there are a lot of people that are less than 60 that die from influenza, and that might be one of the reasons, you know, it's more on people's radar. But I have a feeling coronaviruses after this year are going to be on people's radar for sure. So one of the important features of a pandemic or a virus and its ability to cause a pandemic is how many people it can infect, how many people one infected person can infect. So we call that the R naught, or the basic reproductive value. And this just gives you an idea. COVID-19, for every person that's infected with SARS-CoV-2, 2.5, that person will infect 2.5 people. With influenza, it's about 1.5 people that become infected after from one infected person. And to give you a sense, measles is one of the most contagious pathogens known to man. And for every one person that's infected with measles, 16 more people will become infected. So that's why measles outbreaks are so scary. So on the scale, this virus isn't hugely infectious compared to other viruses, but it is just enough so that it can be propagated efficiently through a population. And that's the main feature we need for a virus to cause a pandemic. The other thing I want to point out is that there are other viruses that infect, other coronaviruses that infect people. So there's this set of human coronaviruses that cause common cold symptoms, and they only replicate in the nose. They don't replicate down in the lungs. SARS-CoV-2 is the new coronavirus that can replicate in both the nose and the lungs. And then the other coronaviruses I mentioned before, the one from camels and the one that happened in 2003 in China, those only replicate in the lungs. So because they only replicate in the lungs, they cannot be transmitted very easily. It has to be a really hard cough. But these viruses are in sneezes, nasal droplets, all that good stuff. So these viruses tend to be transmitted more easily. One of the questions that always comes up is why do some people get severe disease and some people don't get severe disease, and what are the sort of differences? And I know Stu Cohen kind of went over this last time, but this is a cool figure I found, so I thought I'd go over it a little bit this time as well. As I mentioned before, age is a major risk factor for severe disease. People that are less than 10 years old or less than 50 years old, they're most likely to be asymptomatic and have no clinical signs or have mild disease. About 80% of people have mild disease with a little fever, a little cough, your typical, do I have flu? Do I have a cold? That sort of thing. The time for onset disease is really frustrating because it's anywhere from 1 to 14 days. So that because of it averages about 5 days, but because of that, it makes it incredibly hard to contract trace, how do you figure out how long this person's been infectious before they started showing mild disease and evidence of infection that you'd want to contact trace for? And then people that are older, greater than 60, are more likely to have severe disease, and that it consists of dyspnea, heart difficulty breathing, and ICUs may be needed. And this occurs 7 to 14 days after the onset of clinical symptoms. So it's quite a delayed process to progress from a mild disease to a severe disease. And by this time, there's no more virus left. It's from a virologist point of view, a viral pathogenesis guy's point of view, it's super interesting. There's a lot of virus in this phase, in the first 5 to 6 days of disease. And right when you present with mild symptoms, you can find virus, but by the time you have severe disease, the virus is gone, and it's basically the immune response to the virus that is doing most of the disease production, causing most of the illness. And people that are greater than 68 years old are more likely to become critically ill or deceased. About 5% of people that get infected at this age are likely to progress this far. And this is ours. They die of either acute respiratory distress syndrome, acute cardiac injury, or multi-organ failure. And again, this is quite a slow progression. We're out to 3 weeks after the onset of disease symptoms. So it's quite a slow process. And again, age is part of the main confounders in terms of driving the people that are more likely to have severe disease. Again, this is a similar type of drawing, but it emphasizes one thing I wanted to show. This is time down here going out to 14 days, showing critical illness developing about 12 to 14 days, severe illness about 8 days. But the important point that I want to show is that there's a lot of virus early, and some virus when you have mild to moderate illness, but by the time you're out here to 8 to 10 days, there's very little virus. And we're looking at virus genome here. If we look at infectious virus, it would be much less. So it's really interesting to me how we have 2 phases of disease, basically a viral phase, which is almost entirely asymptomatic. And then by the time we get to severe disease, the virus is gone, and it's the body attacking the lungs that is the primary driver of the disease. This is a cartoon showing at a sort of cellular level exactly what that process is all about. And I'm going to go through it pretty quickly and just hit the biggest high points because I know this is more depth than most people are willing to stomach right after lunch. But this is a cartoon showing basically the epithelial cell layer lining the lung or the airways of the lung. We have the virus binding to its receptor and getting in and replicating. We have viral RNA being made, viral proteins being made, a new virus being made and coming out the other side. But the other thing that's happening is we have an innate immune response to that virus. The cell recognizes that the virus is here and starts to undergo an innate immune response, and eventually undergoes a process called pyroptosis, which is basically cell suicide, but it's a special kind of cell suicide that leads to an inflammatory response. So once that happens, we're making inflammatory chemicals here, cytokines, IL-1, things like alveolar macrophages and endothelial cells are responding to that. And the neighboring cells next to this infected cell also respond, making a whole bunch of chemicals that draw immune cells into that area of the lung and try to make an immune response to fight the virus. If those cells are successful and make an effective immune response, then the virus replication goes down. There's very little damage to the lung, and that occurs in about 80% of people. If something goes wrong in that immune response, the immune cells make the wrong sorts of chemicals and the wrong sorts of ratios, then the immune response itself can cause damage to the very delicate cells that form this barrier in the lung between the air and the inside of the body. So when that happens, a lot of fluid leaks out of the blood vessels into the normal spaces that should have air, not fluid, and you basically are drowning in your own fluid. And that's what acute respiratory distress syndrome is. So we prefer to make an effective immune response and prefer to avoid an aberrant immune response, which is going to cause us to have severe disease. So that's basically the difference. When we're trying to develop an immune response, or after we've recovered from our disease, we've made an immune response and we don't want to get infected again. And when we think about vaccines, one of the biggest things we want to look at to sort of guide what we can expect from vaccines and what we should do in making vaccines is to look at the immune response of people that have recovered from the disease. So with SARS-CoV-2, it's been pretty easy to look at the immune response of people that have recovered and sort of try to gauge what type of immune response was responsible for them to recover and how long that immune response is likely to last. And this is a cartoon showing immune responses, response on this side, on the y-axis, and years post-infection on the x-axis. And it shows how long you can expect immune responses to things like the common cold coronaviruses, which I showed you before, that only replicate in the nose. Those immune responses last less than a year. As far as we know, the immune responses to some of the other coronaviruses like MERS and SARS, they last about a year too. If you have severe disease, those immune responses may last a little bit longer, and the T cell responses last longer than the antibody responses. But the bottom line is that with most of these coronaviruses, we do not develop long-lived immunity, and that's in response to natural infection, which is a little bit frustrating when we try to develop a vaccine against a coronavirus that replicates in the respiratory tract. So this is some epidemiologic evidence showing the number of, or the percent positive coronavirus tests in the United States from July 2014 to July 2017. They looked at 4 different coronaviruses. They swabbed this many noses a week, and they they typed the different coronaviruses they found. And what they found was that, like in the winter of 2015, they found OC43, just think of it as the blue coronavirus, and that coronavirus went away until January, the next winter. It skipped a winter and came back the winter of 2017. Why? Because there was a whole bunch of people that still had antibodies this winter, but by the time 2 winters had gone by, nobody had antibodies and the virus came roaring back. The same sort of thing happens with red and green. They are not around in 2015, but they're at high levels in 2016. And then when everybody makes an immune response, they're gone in 2017. Notice the yellow, it's gone in basically 2015 and 2016, and it pops up in 2017 when everybody loses their immunity to it. So these viruses are basically alternating. As you become immune to one virus, then the next virus comes in and takes over, and then your immune system focuses on that, and the immunity to the previous virus wanes, and then that virus comes back, blah, 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 blah. So you can sort of get how the dance goes. And this is why you get colds all the time, and you're never immune, and why we can't make a good vaccine against cold viruses. Now, the question is the same problem going to occur with the deep respiratory coronaviruses? The short answer is we don't know, but it's something we're very concerned about. So let's talk about the development of the SARS-CoV-2 vaccine, which is by far the biggest medical advance I've ever thought possible. I can't believe I've lived through this. So what happened was that the United States Department of Health and Human Services launched Operation Warp Speed last March. It was a partnership between the government and industry that had the goal of delivering 300 million doses of safe and effective vaccine by January 2021. And 99% of all virologists thought this was insane, me included. This ambitious plan, some might say insane plan, initially focused on 125 potential vaccine candidates, but by May 2020, this was narrowed down to 14 candidates. And as of June 2020, this was down to 5 candidates. All this was done through preclinical testing in animal models. All 5 core candidates at that point were all aimed at inducing or designed to induce antibodies to the spike protein, which is what I've been ranting about most of this lecture. So just to compare, before we get to the results, they're saying that from March until December, they're going to get 300 million doses of a safe and effective vaccine. To give you an idea about how long it took to make other vaccines you may have heard of, for polio, it took 7 years. For measles, it took 9 years. For chickenpox, it took 34 years. For mumps, it took 4 years. For HPV, it took 15 years. I've been working on HIV since 1990. We still don't have one. So the average vaccine development is 10.7 years. And these guys, the goal was to shorten this from the usual, they actually said it was 73 months usually, to 14 months. And I again, it's not 14 months, it's been since March. And to break this down a little bit so that people can understand the process, the different colors of these blocks indicate different phases of the process, and those are coded down here. And basically, this is the preclinical phase. When we get into the blues, we're into the phase 1 clinical trials, phase 2 clinical trials. Excuse me, these are the phase 3 clinical trials, phase 1, phase 2, and phase 3 clinical trials. And then 15 months for manufacturing and 6 months for distribution. That's the usual sort of time frame. The process was accelerated by combining preclinical and phase 1 trials. So animal trials and human trials were going on at the same time. Manufacturing occurred from the minute they had immunogenic vaccines in humans and people. Manufacturing, large-scale manufacturing was engaged. And then the phase 2 and phase 3 trials, as we've all witnessed, have been shortened and basically combined to be completed in 6 months. And the distribution process is something we're living through right now. You won't get your Christmas presents, but the vaccines hopefully will be delivered, and that's far more important. So that's the goal, and as we're witnessing, they're reaching that goal, I would say ahead of schedule. They under-promised and over-delivered, which is really none of us thought that this was possible. So this is the candidates that we talk about when we talk about the Operation Warp Speed vaccine candidates. Moderna mRNA 1273 is a messenger RNA vaccine, which will be licensed, at least approved. The EAU, the Emergency Use Authorization, will be approved tomorrow by the FDA. I sat in on some of the meeting today of the advisory board, and the data that Moderna presented was just outstanding. It's an unbelievable, successful clinical trial. You guys are all aware of the Pfizer vaccine, which is another messenger RNA vaccine. It's also was reviewed last week, by the FDA and approved under emergency use authorization. The phase 3s will continue. The phase 3 trial is continuing for that to prove that it will meet all the milestones for eventual biologic licensing. It's not licensed as a full vaccine. This is only emergency use. The same thing will happen with Moderna. The Merck Sharp and Dohme vaccine is still has some safety issues, so it's still in phase 2, but it should start phase 3 trials soon. The Johnson & Johnson vaccine is now in phase 3. It's a replication defective virus, which we'll talk about in just a minute. And the big advantage of this virus vaccine is that people only need 1 dose. With the RNA vaccines that are going to reach licensure or at least authorization for emergency use this week, they require more than one dose, 2 doses. And then you may have heard about the AstraZeneca or University of Oxford adenovirus vaccine, which is also a 1-dose vaccine, and that is in phase 3 trials. And that is undergoing a review in Britain for emergency use authorization, but it hasn't been presented to the FDA yet for an EAU approval. And then finally, there's one more vaccine that was brought into Operation Warp Speed in July, and that is a Novavax vaccine. And this vaccine is a simpler and more commonly used technology. And I'll come back to that in a minute. But this is also now in phase 3, and actually there's UC Davis is participating in this trial. UC Davis also participated in the Moderna and Pfizer trials, as far as I know. So when we talk about these different vaccines, they use very different technology, and it's at least of interest from a scientific point of view to understand that technology, and it may be important from a medical point of view too. But it also explains why we're able to have a vaccine less than a year after the virus entered the United States. So we're basically have a licensed vaccine within 1 year after it was described in Wuhan, the pneumonia was described, and in less than a year after the sequence of the vaccine was published. So these nucleic acid vaccines, there's DNA vaccines that are used in animals, but those probably won't be used in people because the nucleic acid, the DNA, is incorporated into the nucleus and has the potential for actually integrating into the host DNA. So there's a huge safety concern with DNA vaccines. That's the reason why the vaccines that we currently have are RNA vaccines, because those are injected and they're taken up into the cytoplasm of the cell, and they never go into the nucleus of the cell. But once they're in the cytoplasm, as we talked about before, they make protein, and once that protein's made, it's secreted from the cell. And then encounters a different type of cell, immune cell, which is an antigen presenting cell, which is tasked with presenting this viral protein to the immune system so that the immune system can respond. And once that occurs, we get cells that make antibodies and cells that can kill infected cells, which is what we want in a viral infection. The other kind of technology that's being used, and these are the adenovirus vectors that are currently being tested in phase 3, but are not being proposed yet for emergency use authorization. They are viruses that have a bit of the SARS-CoV-2, they have the spike gene of SARS-CoV-2 inserted into the genome of the virus, and that virus is then, it's replication defective, so it's been genetically altered so that it can't replicate completely. It can't make daughter virions, but it can get into a cell. The genome of that virus can be translated into proteins so that protein can be made. And once that protein's made, then it's again presented to the immune system cells, just like the RNA made protein was as well. And then the final way we're going to make a vaccine, or we're testing vaccines, is the Novavax vaccine, which is the last one I described. And that is basically just pieces of protein that are in a syringe, and we're injecting that protein into the arm of people, and it's taken up by the same set of immune cells as all the other proteins that were made through the other modalities, and it hopefully stimulates a similar immune response. So those are the basic formats of the different viral vaccines, coronavirus vaccines that are currently being tested and likely to be brought forward once they get through clinical trials. I have to say that, well, we'll come back to that in a second, I guess. One of the issues with the RNA vaccines, and I saw this in the chat a minute ago, was that they do require extreme care and storage and distribution. There is a difference. The Pfizer vaccine apparently requires -80 storage and can only be stored in a refrigerator for a day and only be stored at room temperature for a couple hours. The Moderna RNA vaccine can be stored in a regular freezer, not a -80, but just a non-regular kitchen freezer, more or less, for about 6 months, and it can be stored in the refrigerator for about 10 days. So there is a big difference in terms of the stability of these two RNA vaccines, and one of our colleagues in chat asked if I know why, and I do not know why. and it hasn't been explained to me. And it isn't being explained to the FDA either, in terms of at the meeting today. Moderna didn't explain how or why they think their vaccine has an advantage in terms of storage, but they did make the point that it does have an advantage. And the question I have is, has Pfizer really tested theirs? If I was them, I would rapidly be testing big lots in -20s and seeing how long they lasted. And they're much smarter than me, so I'm sure they're doing that. So this may change, is the bottom line. Again, these vaccines have only been formulated for about 9 months or so. So we have some learning to do. So what do we know currently from the testing of these vaccines? From the preclinical testing, we know that all of them are safe. In all the animal models that have been tested so far, there's been no untoward outcomes. In non-human primates, hamsters, mice, and ferrets that have been immunized with all of these vaccines, they can all resist infection for 30 to 60 days after they've been immunized. Most vaccine candidates elicit strong neutralizing antibodies and significantly reduce replication in the lungs. It is less clear how many of these vaccines can lower virus replication in the nose. And that becomes a big deal because virus shedding from the nose is probably a major source of transmission. So if people can be vaccinated and still have virus replicating in their nose, that is an issue. Let me back up. Well, let me keep going for just a second. In the early phase clinical trials, all top 6 candidates have proven to be safe, immunogenic, excuse me. And in terms of the early results from the phase 3 trials, we know that the Pfizer BioNTech RNA vaccine was 90% efficacious. And as I said, they were approved last week for an emergency use authorization. We know Moderna is about 90% efficacious, and we know AstraZeneca is 90% in a low dose trial mistake and about 60% protective in a normal dose trial. One of the things that's really important to appreciate about this, I rattled off those percentage efficacy numbers, but what's the definition of efficacy? Well, the drug companies or the vaccine companies get to define that in their trial. And so all of them defined it as prevention of disease, not in prevention of infection. So that the people that are, what they're saying is that these vaccines presented disease in 95% of the people that received the vaccine, but we don't know if it presented infection in 95% of the people that received the vaccine, and it's pretty unlikely that it did. It's probably much less efficacious as preventing infection. Moderna today did present some data regarding the ability to prevent the acquisition of infection. They had 2 immunizations, the first immunization and then the second immunization was 28 days later. And they took a swab at the first immunization. Actually, they looked for antibodies. They made this really clear. They should have looked for swabs, but they looked for antibodies to see if the person was infected before they got the vaccine. Then they got the vaccine. And then at 28 days later, they looked for antibodies to parts of the virus that weren't in the vaccine. And then they judged if the vaccine prevented some of those people from getting infected. And they did find that the people that got the vaccine, there were less infections than in the people that got the placebo. So whether or not that's going to hold up in a bigger trial is not clear, but it is encouraging that the people that got the vaccine seem to have acquired the virus infection less often than the people that didn't get the vaccine. Again, that's not the primary goal of vaccines. The primary goal of the vaccines is to prevent disease, not infection. And most of us think that the virus has to enter the body and then the immune system can act on it. So the idea that the vaccine is a shield that the virus bounces off of and prevents it from entering the body is not something that we generally, not how we conceptually look at vaccines. But it is encouraging, extremely encouraging, perhaps, that the amount of virus that could replicate once people that got the vaccine became exposed is so small that they didn't make an antibody response. So anyway, that's very promising. As I alluded to a minute ago, the Pfizer vaccine was actually approved 2 weeks ago. We were 1 week late. We were on the 17th, so we were on the 10th, and Britain was on the 4th. The first people that received the vaccine in Britain were on December 8th. And one of them was classically named William Shakespeare. I'm sure you guys all saw this on the news too, but I thought that was pretty cool. So as I alluded to all throughout this, December 10th, the Pfizer EUA approval was recommended, and on December 11th, FDA approved the Pfizer vaccine for emergency use. As you probably all know, Tuesday, people at the Med Center, the people at highest contact with COVID patients, started being immunized. Yay, things are rolling. The Moderna vaccine, we expect to be, the committee should have adjourned by now, or is close to adjourning by now, and I'm sure they recommended approval, and then the FDA will make the official approval tomorrow, we all assume. And then the Moderna vaccine will go into people's arms again about 4 days after that. So by probably around next Tuesday, the Moderna vaccine will start going into people's arms. So great, the pandemic's over, we got vaccines. Well, sorry, that's not really what's going to happen. So this is the best guess of the epidemiologist to the effect these two vaccines with 98% efficacy are going to have on the pandemic over the next 6 months. Here's the current graph, and it shows you where where we're starting the vaccination from. And because we're starting here, it means that we're going to have 10 million more people infected by the time June rolls around. This is over the next 6 months. Sorry about my dogs. And we'll have about 160,000 more Americans will die over the next 6 months between now and June. So the vaccines are great, but people are people and they need to mingle. And because of that, we're starting at a very high place with this vaccine program. If we would have started down here, we would have had a much fewer deaths and many fewer people infected. But it is what it is, and we're doing great in terms of developing a vaccine this fast. I really can't overstate how amazing it is to have watched this process from afar. I'll have to say that I do feel, and most academics feel, a little left out by this whole process. The first animal studies that were done in China were started in December of last year. So they isolated the virus in mid-December, and they had it into monkeys in December before the end of the year in 2019. The first monkey studies in this country were done in February of last year. And we didn't know about them, and we didn't have access to the virus or any money to do the studies. My lab was able to isolate virus from the first patient, and the primary goal of that at UC Davis, and the primary goal of that was to have RNA to provide the clinical labs so that they could develop diagnostic assays. But the secondary benefit was that we were able to have virus by the end, mid-March, and so we were able to start monkey studies in April on campus here. But that's still too late, although most of the preclinical testing had been well underway by then. So anyway, that is the sort of story of where we are now, and hopefully where we're going to end up once the vaccines are rolling. And I'll stop there and jump into the chat and see what the questions are. Is there any speculation on how long the immune response to an mRNA vaccine will be active in an individual? No, I mean, there's lots of speculation, but nobody has the data yet, and that's certainly a major goal of the long-term follow-up to these trials. So that we'll know in a year, and hopefully people will still have immune responses in a year. But yeah, it's going to be interesting to find out. Anonymous attendee, a coworker expressed concern regarding COVID vaccines and women of childbearing age alluded to potential placental issue. Is there any information about this? I'm not aware of any placental issues. I mean, in general, you don't want to excite, I mean, first of all, it's best to do actual testing, safety testing in that setting and make sure that they're safe. But most vaccines are safe in pregnant women, and some of them, as I'm sure many of you will know, are recommended for pregnant women so that their babies aren't exposed during the birth process. But I haven't heard anything specific about any potential for placental issues with these vaccines. Is there any supported evidence that people immunized against MMR have some protection against SARS-CoV-2? Something I read about that was theorized about why children may be asymptomatic. So this is a question regarding whether non-specific immune responses could confer some protection against SARS-CoV-2. And the bottom line is that we don't know. It's not just the MMR vaccine, but people have also speculated that the BCG vaccine, which is a vaccine used against tuberculosis in almost every country in the world except ours, and it's given orally to children. It cranks up their immune system, and that's also maybe it's been epidemiologically hypothesized that that could be associated with resistance to a number of different pathogens and perhaps SARS-CoV-2. But we don't have data. It's an interesting idea and one that's being pursued, though, and that's why I brought it up. I think there's 4 randomized controlled trials that are going on right now to test the hypothesis regarding the TB vaccine, but I don't think there are anything like that with MMR. Let's see. Do we know the half-life? Yeah, no, it's the same question. Peter Pasco wants to know, do we know the half-life of the messenger RNA in the cell? It is short-lived. And what regulates the half-life of the messenger RNA? Is the same thing that regulates normal message RNA in a cell. So I think that's what Peter asking. So there are a bunch of regulatory mechanisms, including microRNAs, which control how much message RNA is in a cell and how long that message RNA lasts in the cell, and if it's effectively translated on a ribosome. And there's a lot of regulatory control of messenger RNA. What the Moderna guys said, and there are some animal studies that have looked at how long a synthetic RNA injected into the muscle lasts, and it seems to be very short, in mice, a day or 2 at the most. What the Moderna guys said, though, today was they showed a cartoon of a human and a cartoon RNA going into the arm, and they made it sound like it was hours. But there's I don't know of any data that says that. So I would stick to the mouse data that says the mRNA, the foreign mRNA that's in the vaccine lasts for at most 48 hours, but more like 24 to 36 hours in a cell. And I hope that answers the question, Peter. William Pevick, why would infection with the virus be expected to provide less immunity to the vaccine? Yeah. So William Pevick brings up the classic question of why would infection or a vaccine provide better immunity than the virus itself? And we don't expect that. We expect that a real viral infection should cause better protection. So that's why I showed that data related to the longevity of the immune response to a viral infection. With these coronaviruses, it's relatively short-lived, a year or 2. So it may be that the mRNAs, vaccines, and the other vaccines only elicit immune responses that last for a year or 2. We really don't know yet. The antigens are being presented different ways, with different sorts of cells being interacting with the antigens for the first time. So these are all really good questions. We're going to learn a lot from studying these people. And then, yeah, Sally asked the next question, are there other mRNA vaccines already in use? Absolutely not. There are no there are no RNA vaccines in use in humans until now. There are no DNA vaccines for use in humans. There are no live virus vector vaccines. So there are no recombinant vector vaccines for use in humans. So all of these things are new technology. The only one with sort of the standard technology is the Novavax vaccine, which I talked about, which is just protein. And that is a technology that's been used before. And Novavax actually makes a licensed influenza vaccine. So that is well tested. The advantage of the RNA vaccines, though, is that all we need to make them, all the companies needed to make them, was that sequence of the virus, the spike protein that was published in early January last year. So as soon as that sequence was published, they could start actually making real vaccine and scaling it up and putting it into animals. So they had a huge advantage over people that had to make the RNA and then express the RNA and then make the protein and purify the protein and make sure the proteins have the right conformation, et cetera. So that's the real advantage of the RNA vaccines. We hope they're going to work because there's another pandemic coming, and these things are so cool and so high tech and so easily manipulated that we really, really hope that the RNA vaccines are going to work. And so far, so good. It's like I said, I'm not involved in any of these vaccines. I test a few RNA vaccines, but it's just beautiful technology, and it's so easy to scale up and shift to new targets. All right, if someone is on an immunosuppressant drug for RA, are all of the vaccines safe, or some safe or not safe? First of all, we don't give medical advice over the Zoom, so you always talk to your primary care physician. But in general, none of these can replicate, so none of them have the potential to propagate in an immune suppressed person. So the RNA is not going to make more RNA. The adenovirus vectors can't make more adenovirus vector. All they can make is the protein. So in general, you should be safe, but always check with your primary care. What could we have done to prevent being the country with the most COVID cases? Seriously? The usual stuff, masks, social distance, wash our hands, that sort of thing. And do it with gusto. China is the example. Wuhan, that's the extreme. We can't do that in this country. But if we could have Wuhaned, we would have had a lot less cases.
Dr. Miller,
Question.
Julianne. So
Go ahead.
we want to be conscious of your time. It's
yeah.
Before 2:00.
I didn't keep track of the time. Yeah, I'm okay. If you guys want me to go to the end, I can go to the end of this list, I think, without any problem.
Okay, well, if you're comfortable with that, I'm comfortable with that. And if people need to leave, they can sign off. So you're doing a great job. Thank you so much.
No problem. These are all, totally reasonable questions, and everybody has them. So I have no problem answering. Some reports indicate that Moderna is less effective in older adults than Pfizer. I haven't seen the data, John, so I can't give you any rational thoughts on that. See, those are easy questions. When will the AstraZeneca vaccine be approved as well? That's a tricky question. They have some wrinkles in their data that they need to do a good job of explaining, and so they're taking time to get their story really straight before they apply. The thing with the FDA is that you get basically, one chance to present your data, and you want it to be the best it can be. So they have to understand what happened when they lowered the dose of vaccine. They got more protection, and they need to understand that and be able to explain that. And that was a mistake when they lowered the dose of vaccine. When can we expect the seniors get the vaccine? Yeah, it's a big question. And by the way, I want to be in that category. That's a really good question, Barry. Nobody knows for sure. It's all related to the supply chain. Realistically, if you're not vaccinated by April, May, June, I'd be disappointed. But we'll see what happens. If people would quit buying stuff from Amazon, then we get this stuff shipped around faster. So there's just so many variables involved. The next supply chain issue is going to be syringes and inject needles, and then humans that are trained to do injections. So there are going to be some potential glitches. Company says it's 95% effective. That means that far fewer people get the disease compared to who received the placebo. Yeah. With infection rates of 30 per 100,000, that means 10 people will get the disease. I don't know if it, yeah. So the actual numbers are that less than 200 people got infected in the trials. And when they broke down who in those 200 people in the phase 3 trials for both of these RNA vaccines, less than 200 people got infected. With Pfizer, it was around 100. With Moderna, it was around 175 or something. And when they broke that down, 95% of the infections were in the placebo group, and only 5% were in the, and 95%, when we say infections, we mean diseases, it's clinical disease, was in the placebo group, and only 5% was in the vaccine group. So that's what they mean when they talk about 95% efficacy. Will that change when they add more people? Absolutely. They do have 30,000 people enrolled in each trial. Right now, I think the numbers are coming off of like 10 to 15,000, something in that range, Peter. So when they get into the full trial, will this change? I think it will change. Yeah, but still, the statisticians say it won't change that much. Maybe 4 or 5% in probably the negative direction, but it's still pretty amazing. So that's how they make the conclusions about efficacy. Any thoughts or comments on the people with anaphylactic responses to the Pfizer vaccine? Yeah, that is scary. But you know, every vaccine has the potential for an anaphylactic reaction. Anytime you inject any foreign material, you know, usually a protein into a patient, you have the potential for anaphylaxis. It's part of the challenge of vaccines that we're treating people that are healthy and you're trying to not do any damage. So yeah, it's a little bit unnerving, the anaphylaxis stuff, but it's not, you know, unexpected, let's put it that way. And I don't think it's out of line with what's been seen in other vaccine trials. Should those who've been infected get vaccinated? Heck yes. Did you see that data I showed you about the vaccine waning? I mean, the immune responses to natural infection waning. Yeah, I think at the end of the day, we're going to be getting coronavirus vaccines probably every year or maybe every 2 years. It'll probably be like a flu vaccine, and possibly, you know, every 2 years. And it might start shifting. So the virus might, once we get herd immunity, then the immune responses of the herd might make it so the virus has need to change its spike protein a little bit, and that could lead to escape from the current vaccine, and then we'd need a new vaccine. Yeah, so that's the point. We'll have to make new vaccines. Diane wants to know, but I think the beauty of like the RNA vaccine is we'll just sequence the new virus, switch out an amino acid or two in the sequence of the RNA vaccine, we'll have a new vaccine that's ready to go. What do we know about the ACE2 receptor? Can it be inhaled, an ACE2 agonist? Yeah, so there are a lot of people trying to test ways to block ACE2 receptors in the respiratory tract as a therapeutic approach to, or even a prophylactic approach to preventing infection. But nothing has sort of hit the literature or any of the meetings I've been attending by Zoom indicating that that's a successful approach. But, you know, a lot of people are trying it. Let's put it that way. Oh, looks like we've reached the end, you guys. You happy?
Very much so.
Okay, I'm happy to, you know, answer the questions, and I hope everybody found the presentation useful.
So, Dr. Miller, thank you so much for this. Obviously people were very interested, and we had 145 people attend, which is amazing. So we're really excited by this. And if we get any questions from the audience, they can certainly email the Retiree Center. And if it's something maybe we think we can send on to you, we will. But I think you answered almost all of those questions, and we all learned a lot. So thank you for your time, and we wish you well. Stay healthy, and happy holidays, and happy holidays to everybody.
Same to you guys. Thanks a lot.
Right. Thank you so much.